Professor Kasira is the director of the Controlled Environmental Agriculture Center and a professor in the Biosystems Engineering Department at the University of Arizona. He received his bachelor degree in agriculture engineering in Corkova University in Turkey. And he obtained his master's and PhD degrees from agricultural and biological engineering from the Ohio State University in the USA. So this is my alma mater. I got my PhD from there. Probably, he was an all-star, and I got my PhD from Columbus, Ohio. Anyways, his research involves automation, environmental control, alternative energy integrated sea systems, and the resource use optimization and controlled environmental agriculture systems, including greenhouses and vertical farming-based factories with artificial lighting. So this is really quite impressive. So, and he's a member of American Society of Agriculture and Biological Engineers and the International Society of Horticulture Science, ISHS. So he serves also as a chair of the Division of Precision Horticulture Engineering under ISH. So quite really accomplished leading scientists in this field, and we look forward for his talk. And I would ask every one of you, please, to give your full attention, and please turn off your microphone if you didn't do so already. And we look forward to Professor Murat talk. Then after that, Professor Mark Tester will take over to present his talk. And Professor Murat, so you can now start your talk.
Thank you very much, Magdi. I really appreciate that kind introduction, and go Bucks. Nice to see an alumni in Buckeye there, but also go Wildcats as well. And thank you very much for the invitation and for having me for the summer seminar series. I appreciate the invitation from my colleague and friend, Dr. Rod Wing, and the KAUST team. I am super excited about this opportunity to have a conversation about our research programs, our experiences, and exploring the possibilities towards future collaborative opportunities to form a joint center for sustainable agriculture in desert environments. I'm joining you today virtually. However, I had the pleasure of visiting KAUST campus twice, back in 2011 and 2012, when there were discussions about forming a desert agriculture research initiative under the leadership of Dr. Nina Federhoff. She was assigned there at the time, the CDA, to lead this discussion and this opportunity. So we were invited as part of the team for brainstorming ideas. What are the possibilities for KAUST at this location, and also forming an international kind of collaborative team and opportunities. As you can see, we were having discussions there, and the Dari site was actually envisioned to be right across the beacon, a beautiful site back then at the time, considering greenhouse technologies, low tech to high tech, solar technologies, photovoltaics, cooling systems, dehumidification systems, water desalination, and also cafeterias to enjoy the view and the site. So those were kind of the discussions we had back then. And considering our climates, there are similarities between TWAL and KAUST and also Tucson, Arizona, but also there are unique differences in our climates. We are in arid desert climate, as you are located in a coastal desert. So your temperatures, as you can see, the solar radiation and the air temperature throughout the year is really condensed in one zone, whereas the climate in Arizona, because of our arid desert climate, the day and night temperatures can be dramatically different depending on the season. Humidity is a factor for us during the monsoon season. However, in your location, the humidity is a challenge throughout the year. So the discussions also focused on identifying technologies that could dehumidify and cool the greenhouse and controlled environment agriculture spaces, using chilled water technologies or liquid desiccant systems, so we can provide the optimal conditions in those controlled environment growing spaces. Very primitive systems or maybe locally available resources, from pozzolan bricks to really high-tech systems discussions. So considering the climate change and the challenges we're seeing in terms of the resource limitations, the arable land degrading or becoming less available, and access to water, labor, and energy is becoming challenging, we will need to have integrated, complementary, and smarter production agricultural systems. We will continue to grow staple crops in field conditions. However, I believe controlled environment agriculture will have a significant role to feed the populations and provide them fresh, nutritious food in a safer way and in a sustainable way, considering greenhouses, for example, natural sunlight, using natural sunlight, or maybe in indoor agriculture spaces using artificial lighting, such as those with vertical farming systems. But also there will be opportunities for alternative proteins or products, maybe mushrooms or edible insects, aquaponic systems as circular systems, integrated systems to leverage resources, maybe convert waste into usable resources and inputs in these integrated and sustainable systems. When it comes to controlled environment agriculture, there are different levels of technologies available. There are low technologies such as hoop houses or high tunnels, but medium high-tech greenhouses. These greenhouses could be on the ground, or it could be also located on rooftops in urban settings, but also highly controlled, confined environments such as with vertical farming technologies. So I want to say that there is no single silver bullet solution when it comes to identifying the technology. It should be adaptive technology, adaptive approach to meet the expectations of the production outcomes in terms of quality and quantity, but also local needs, the consumer needs, available resources, finances, and also capabilities of the users of these technologies, from production side of things to engineering and technology side of things. So we have to consider all of these factors when deciding and integrating these controlled environment agriculture systems. So why controlled environment agriculture? I have a list of reasons to consider controlled environment agriculture. As you can see, production quality and consistency is one of the significant one. We can achieve consistency and quality in greenhouses as well as in vertical farming spaces and technologies. Water use efficiency, resources efficiency is greater when we consider greenhouses and vertical farms compared to field-based agriculture. But also there are other roles, other factors that affect the decision for controlled environment agriculture. And I also want to underline this one on the list. It really captures the imagination of young generation. They are technology centric, they are data centric, they are interested in ag tech and new technologies, innovation in agriculture. So that is really attracting the young minds and young generation towards controlled environment agriculture. The bottom line for any kind of agricultural system is the resources efficiency. We have to consider how much resource we are using, what are the level of resources we are using for the production, and how much outcome or production outcomes we're achieving for that from that production system. When it comes to controlled environment agriculture, on the input side, we have energy, electrical energy, water, carbon dioxide, fertilizer, and labor as inputs. And these are provided for the plants and the plant environment. And then on the outcome side of the list, we have edible biomass, but we also have non-edible biomass, which can be converted into resources. They may become valuable inputs in a circular system and recycling systems, and we can achieve these recycling of resources such as water, nutrients, for example, within controlled environments, but also transpired water from the environment. This water can be harvested from the air and brought back into the hydroponic system in unique ways so we can improve the resource use efficiency. So the fundamental difference controlled environment agriculture and field agriculture is that we have almost a complete control on both demand and supply conditions in controlled environments, being able to convert some of the waste materials into useful resources. However, understanding the plant microclimate interactions are needed to develop resources efficient, resource conserving control and management strategies and practices within controlled environment agriculture systems. The controlled environment agriculture systems and processes are complex. And here's an example of that. Complex processes interacting with each other in a greenhouse example. So we must consider the biology, the plants and plants requirements, and combine that with the greenhouse physics, the physics of the environment, so we can achieve the greater potentials, we can achieve optimal production and high quality production, resource conserving production practices in controlled environment agriculture systems. Plants must be the center of the attention. We have to develop the environment, develop the systems and technologies around the plants and plants requirements. From an environmental control point of view, it is a better approach to consider plants as sensors to communicate their needs so we can maintain the conditions, we can deliver the conditions from the environmental control systems in a resource use efficient way. And further to that, if we can provide and enable the users and operators with data, meaningful data, and data platforms, then we would empower them so they can achieve the highest capabilities and optimal growing conditions, resource conserving conditions in controlled environment agriculture systems. Automated crop diagnostics, decision support systems are among emerging automation technologies with computer vision, machine vision is being considered. So these systems provide direct information from the plants, phenotypic information related to these can be related to plant health and growth, then using this information back in the system for decision-making or control purposes. So again, these are the trends that we will be seeing more integrated into controlled environment agriculture systems. And we have been working in this area in our research programs with computer vision, robotics assisted plant phenotyping and plant health and growth monitoring using textural, temporal information, multispectral information to understand the growth and to detect anomalies in a controlled environment agriculture production setting so we can take timely actions to correct these anomalies in a more resources efficient, more sustainable way. So considering plants in addition to the aerial and root zone environments would be the best approach, to towards optimal and sustainable production systems in controlled environments. Robotics is another developed emerging area in terms of their integration into commercial greenhouse systems, because there are many repetitive processes that enables the robotics systems integration into greenhouse or controlled environment agriculture systems. The challenge so far has been the speed of processes in terms of harvesting, maybe crop management practices. However, now with the technology advancing, the cost is being reduced, as well as the canopy architectures or greenhouse system designs are advancing in an innovative way, we will see further integration of robotic systems, both in greenhouses as well as in vertical farming space. There's a lot of interest in vertical farmings too, considering warehouse-based facilities or maybe shipping container-based systems. The attention here is the capability of producing greater amount of yields within a smaller footprint in this multi-tiered stacked hydroponic systems, especially considering urban settings and the land availability, the cost of the land, and also with the interest of providing the producing and providing that produce to the market within a close distance to the marketplace is attracting investors and also those who are interested in vertical farming systems and technologies. Here, I am sharing some of the systems, larger installations in the United States, but you can expand this list to other places and regions around the world. They are also right now connecting with traditional agriculture based companies to grow their produce in a more consistent way. The challenge, again, now that we are seeing from the traditional growers is the quality and the consistency, the flavors that are coming from these products and produce. So, and also the inconsistency in terms of the climate in different locations, again, water resources availability, the quality, labor availability are challenging the traditional growers. So they are interested in investing technologies for greenhouses as well as vertical farm in an integrated fashion, again, both field-based agriculture and controlled environment culture complementing to each other to meet their expectations, market demand and consumer expectations. However, there are a lot of challenges in vertical farming space as well, mainly with the electrical energy use for cooling, dehumidification, and also for lighting needs in vertical farming space. So we have this a multi-state project funded by USDA, NIFA, optimizing indoor agriculture for leafy green production with an acronym of the Optimia. We're collaborating with our colleagues from Michigan State University, Ohio State University, and Purdue to develop production technologies, systems, practices that will enable us to reduce the energy use as well as improve the quality, the flavor of the produce grown in vertical farming space. Our contribution from my lab here from the University of Arizona is emphasizing the innovative environmental control applications with algorithms that will be developed to maintain or manage the lighting schemes as well as other resources, or in a better way to describe this, co-optimizing environmental conditions so we can achieve resource use optimization in vertical farming space. Again, here we're really focusing on the capability or the ability of manipulating or controlling one of the variables while controlling the another one in a way that it leads to energy and resource savings. Here's an example of a research that was conducted in our lab by one of my former graduate student, Brian Kaplan, with the focus on daily light integrals, the light intensity and photoperiod, or total accumulated light delivery in a certain range. Here, as you can see, low daily light integrals from 9 to 19 DLI levels versus enhancing CO2 levels or changing CO2 levels to determine if there's any energy saving potentials. But here, our focus was on the electrical energy use efficiency. As you can see, depending on the daily light integral levels provided, so we are able to, yes, to produce higher yields under high light intensities. Of course, that brings other several challenges, as we use lower light intensities or data light integrals. As you can see, the electrical energy is reduced, but at the end, the electrical use efficiency is almost the same. So in this case, from a production viewpoint, one should consider how much electricity we are using and the cost of that versus the quality of the produce. If that is within the desirable range, then some strategic decisions should be made and integrate these decisions into environmental control strategies. In a vertical farming space also, in a technology, because of the limited headspace between the LED lighting and the plant canopy, the challenge is the lack of sufficient airflow and airflow uniformity, leading to non-uniform environmental conditions in that limited headspace. So because of the stagnant boundary layer, the photosynthesis is affected in a negative way, transpiration is a challenge. So with leafy greens such as lettuce, most of the time tip burn becomes an issue. This is the lack of calcium being transported to young shoot tips, in turn burning of the tips, leading to unmarketable yields and qualities that may prevent or challenge the grower in terms of marketability and profit. So using air distribution systems that can deliver the airflow in an effective way to the canopy in a turbulent airflow fashion would enhance the airflow and also the transport mechanisms within the plant, reducing the boundary layer thickness, enhancing the stomatal conductance, enhancing the transpiration, leading towards more optimal growth and higher quality production, as well as optimal resource use in vertical farming space. So we have been investing time in this, and our emphasis with research has been more experimental as well as simulation modeling based approach to design, to first evaluate the existing system designs to understand the uniformity within vertical farming designs, and then identify alternative approaches to improve air distribution capabilities or enhance uniformity of the environment under different scenarios, with what-if scenarios for vertical farming applications. And some of these designs that we created and presented to our industry and industry collaborators are already in place and being used in commercial settings. And some of these information and research outcomes are presented in our publications, as well as in this textbook book called Smart Plant Factory. Second one will be published very soon, so I encourage you to review the information we're presenting if this is an interest to you. We've been also doing research on energy use efficiency and comparing the greenhouses versus vertical farm. And one of my former PhD student, Ying Zhang, did research in this area, considering different locations, climatic regions around the world, for example, with Riyadh, Abu Dhabi, Miami, Phoenix, Seattle, and Duluth. Each of these has unique climates, from hot, dry to hot, humid to very dry, very cold locations. So our interest was to identify the electrical use, energy use efficiency between the greenhouse as well as vertical farms, and also with, again, what-if scenarios in terms of technology integration or the way the technology is used and operated through environmental control systems, leading towards energy saving potentials. So in this first graph here, you're looking at the comparison between the electrical LED lighting system efficacy, the amount of electrical energy versus how much emission, photons emission we achieve from LED lighting system. Of course, this affects significantly the amount of energy used, electrical energy used within the vertical farming systems and technologies. As you can see, with two efficacies or three efficacies for LED lighting system for different regions are presented here in a vertical farming system and design. And you can also see the difference in comparison between the energy used for lighting, cooling, and heating applications. Another what-if scenario that we evaluated in this research was to look into the number of tiers from 2 tiers all the way up to 11 tier systems to understand the energy consumption, as well as using economizer modes with heating, ventilation, air conditioning systems in a way that if the outdoor conditions are favorable, the idea is to use the outdoor climate and conditions in a unique way so we can reduce our electrical energy use for the air conditioning applications in the vertical farming system. So as you can see, the comparison here between non-economizer versus economizer mode, 2-tier system versus 5-tier system. As you can see, we're able to achieve some energy savings, electrical energy savings, when we consider those operational modes. Finally, our comparison between the greenhouses versus vertical farming systems. Again, this is all based on lettuce growing in hydroponic systems. As you can see, in warmer zones, in locations when the light intensity, light is available and abundant, greenhouses are slightly better in terms of electrical use efficiency compared to vertical farming systems. However, I just want to point out that the cost of resource in terms of electricity and water is different also when you consider the locations around the world. In one location, maybe electricity could be cheap, accessible, whereas the water accessibility might be difficult or challenging or limited. So again, when we're deciding on technologies, we have to consider these factors and resource availabilities and accessibilities. I would like to see lighting systems controlled by plants, using plants as sensors and mechanisms to control the lighting dynamically in vertical farming systems as well as in greenhouse systems. So in our lab with my student Casey Shashteen, we are working on developing photometric feedback-based decision support and control applications for vertical farming systems. And this research integrates modeling-based approach to predict the plant biomass and plant growth in relation to environmental variables such as air temperature, light intensities, the CO2 availability, to identify, to determine the growth and use that information as part of the environmental control so we can control the operation of lighting currently with light intensity only, but towards also in the future being able to control the spectrum in vertical farming space. So we're also looking into energy saving potentials and capabilities. What if scenarios such as we dim the light a little bit while alleviating the CO2 to be able to achieve the same level of produce yields, and what would be the energy saving potentials. In the graph here on top right corner is showing you one example of that strategy as we deviate from 500 ppm to 1100 ppm, and then working between 13 versus 11 daily light integrals as part of the control strategy. As you can see, a 14% energy savings is achieved through that strategy. But we're also curious and trying to understand if there are potentials for carbon dioxide use and savings in CO2 in vertical farming space. As you can see here, we're also trying to determine what would be the production yield and outcome if we dynamically control the CO2 during the growth phase of these leafy greens in vertical farming systems. And this is currently a modeling-based, simulation-based study. However, we also just concluded our experimental study, so we are comparing the simulation versus experimental data obtained to develop strategies for control applications. Plant phenotype is a function of its genetics, environment, and management. So the GEM, we can focus on environment controlling the environment through the technology, the engineering approach. The management can come through the operators and operators' capabilities in conjunction with the technology used, environmental control systems, but we have to consider the genetics, the genetic potentials, new varieties through breeding. So we have crops and crop varieties that are more suited for use in controlled environment agriculture, as well as being able to use the resources under resource limiting conditions or stressful conditions, for example, water stress or nutrient stress, so we can achieve or realize the full potential of the crop and production outcomes. So there are great potential. We have to invest time and interest into pushing plants out of their optimal and comfort zone in a way that we are still able to achieve acceptable yield and quality. However, with that approach leading to resource savings or optimal use of resources, those limited resources in controlled environment agriculture systems. There is a lot of interest in terms of horticultural lighting with LEDs, but there are so many questions that we need to also address. I'm not going to go through each of these. They are listed on the slide, from design, their integration, their operability, the light use efficiencies, the system designs to be able to deliver the light effectively to the plant, as well as, again, breeding to enhance light use efficiency. So we can do so much with engineering. We have to combine that with the biology and genetics improvements so we can realize optimal outcomes. We are looking into circular systems. Again, as I mentioned before, being able to leverage a waste stream and converting that into an input resource in controlled environment agriculture systems. And here's an example within the project that we are working together with my colleague Better Pryor here from Plant Sciences, as we are interested in the CO2 emissions coming from mushroom systems as a waste or ejection from that system, so we can leverage that CO2 and be able to use that in a growth system, a growing system for leafy greens, and then maybe consider the humidity from that environment or the water capturing from that environment and utilize that for the mushroom side within this system. So we're really interested in circular systems, recycling systems towards sustainable crop production in vertical farmings. Again, we'll see more automation, robotics in vertical farming, a lot of interest in developments in Internet of Things and cloud-based services and data management, augmented and virtual reality to connect the biology, the virtual environment with the physical environment, providing unique experiences and capabilities for the user is technology trend, and we'll see more integration of these technologies within controlled environment space. Artificial intelligence. Can artificial intelligence do as well as a farmer in the future? Maybe for certain types of systems, but I don't think that AI will replace the green thumb and the operator and the user. I believe the AI will enable and empower the user, the operator, so we can achieve greater potentials for yield as well as resource use in controlled environment agriculture systems. I have always been interested in alternative energy systems, specifically with photovoltaics. Looking beside a greenhouse and always thinking about why don't these greenhouses produce their own energy. I think we need to focus more on agrivoltaics, being able to co-produce the food as well as electricity within the same footprint. The slide summarizes some of our research activities since 2012, considering traditional silicon opaque photovoltaics, running greenhouse systems in an off-grid mode, growing different kinds of crops in lower tech systems, medium tech systems, but also we evaluated and studied luminescent solar concentrator based wavelength selective photovoltaics to explore the possibilities, again, both capabilities and limitations towards improved integration within controlled environment agriculture systems. Recently, we just completed a project that focused on organic thin film plastics, organic photovoltaics technology, where the interest is the wavelength tunability, as well as electricity production capabilities in controlled environment agriculture. Here's an image of that greenhouse and research on this slide. We just published actually a paper on this, so if you would like to see more details about the research outcomes, as you can see here, the graph on the lower left corner is presenting the transmittance, as you can see with this OPB technology we used, transmitting some of the light within the 400 to 700 nanometer photosynthetic active radiation range. And again, also the near infrared portion is somewhat penetrated, transmitted, and relative to the photosynthetic efficiency, as you can see here, in terms of the transmission of the light. But you can see here the cumulative yield achieved after transplanting date between OPV integrated greenhouse versus controlled greenhouse using double layer polyethylene film. But at the end, when you consider the light use efficiency with the cumulative light in the photosynthetic active radiation range received by the plant versus the cumulative yield outcome, you can see a greater light use efficiency is achieved using organic photovoltaics technology and film. Of course, we also observed some limitations and challenges with the current organic photovoltaic systems and technologies because of their low efficiency, energy conversion capabilities ranging from 1% to 3% range, and also degradation. Burn-in time is very short. So these are, or also deployment strategies must be considered depending on the crops being considered for production. But these are all great research questions. So we have to look into enhancing these technologies so we can have energy producing greenhouses in the future. So this research was part of my graduate student, Rebecca Waller's PhD research, and she successfully defended and she accepted a position at KAUST. And I am happy to share that news. She will be heading your way there. And I believe her joining to KAUST team, I see that as a kind of initial steps also towards our future collaboration and opportunities. Off-grid greenhouses, photovoltaic systems, integrated controlled environment is also part of our National Science Foundation funded project, as we are collaborating with our indigenous communities here in the state of Arizona with Navajo Nation. We are considering off-grid greenhouse systems for food production, off-grid nanofiltration systems for water treatment, because indigenous communities, especially Navajo Nation here, are challenged with access to potable water and electricity. 30-35% of the nation does not have access to water as well as at same level to electricity. So we are developing these technologies. We are educating, training next generation educators of the indigenous communities so they can take these knowledge base into their communities and address these challenges. So we're collaborating with 7 academic units, engineering units here under the College of Engineering within this project. But we are also considering other technologies, unique systems, for example, quantum dots as optimizing spectral quality with special films that can be used as a covering material in controlled environment agriculture systems. We are investing our resources and interest into mycology, again, alternative proteins with mushrooms, nutritional enhancement, as well as automation to achieve greater yields and qualities with mushroom production in controlled environments. Food waste, using food waste and converting that into proteins and edible insects is another research program that we are working on. My colleagues here, Gogi Davidovitz is leading that research effort. So as we are focusing on earth-based systems to innovate and to create sustainable agriculture systems with controlled environments, we are also interested in space horticulture. We have historical progress. We have history working in bioregenerative life support systems and system designs for surface applications on Moon and Mars as bioregenerative life support systems. We are currently funded through NASA as we are developing efficient water and nutrient delivery systems for crop production in the microgravity environment of the International Space Stations. So there are challenges that we need to address in that space as well. So we are very excited and trying to use our experiences and capabilities from the Earth-based controlled environment agriculture systems to space-based agriculture systems. I just want to say this too. We are so proud of our graduates going through our programs. They are out there in the industry doing great things and having huge impact and becoming leaders of the controlled environment agriculture. So this is one of the reasons that I am so excited, and this keeps me coming back to my office and try to keep working with them and try to shape the future of controlled environment agriculture together with our students, staff, and volunteers and our faculty and colleagues. So with this, thank you again for the opportunity. I hope we have some time left so I can maybe respond to those questions. Thank you.
Wow, this is really impressive. Thank you, Murat. This is really impressive talk, impressive advances, technologies, and definitely these will undoubtedly reshape the future of agriculture. This was really quite impressive talk. So guys, questions, please. Questions?
Hi, Magdi, I've got a question.
Yeah, sure. Go ahead.
So, yeah, this is really nice stuff. I really like the, like the energy balance and dynamics also. Is there any kind of assessment on looking at the energy use and the carbon emissions, comparing like an on-site controlled environment production versus, far away production in some other location, and then the shipping, energy costs that go into, like a field production, but then you have to ship it either ground or air freight a long way away.
Thanks, Jesse.
How do those compare, right?
Yeah, that's a great question, actually. I shared with you some data and information we were able to generate from our research comparing greenhouses versus vertical farms, not comparing that to field agriculture right now in terms of energy use, electrical energy use for different locations and climates around the world. But the life cycle assessment and data and information comparing greenhouse to vertical farms to field-based agriculture is limited, is lacking. It's something that we need to actually work on. And to my knowledge, there's only one research, multi-state research project funded through NSF at this point, kind of focusing on that. So I don't have any data that I can share with you at this point, but there is a great need to be able to understand, the carbon footprint and life cycle assessment between these different kinds of agricultural production systems. So we can improve them and identify ways to, improve each of these from the greenhouse to vertical farm to, as well as the field-based agriculture.
Great, thank you.
Thank you. Any more questions, guys? Questions? Okay, so if no one has a question, so I have just one question for you, Murat. So you have been here like almost a decade ago, right?
Yes.
I know that you have been familiar with dairy and the ecosystem here and what we wanted to do. So what is possible today that was not possible 10 years ago? So in that ecosystem, I mean, something specific for projects similar to dairy.
Yeah, I really, it was a privilege to be on KAUST campus, a beautiful campus, and then with, faculty, stellar faculty there, and students and staff, and capabilities, and also a lot of challenges, as I showed on like the graph there, the temperature, high humidity, high temperatures, and that really challenges the production, not only in field agriculture, but also within controlled environments. I think in terms of technology-wise, 10 years ago, we had similar technologies and systems. I think there are now new capabilities, advanced capabilities in terms of environmental controls, in terms of data analytics, and also, further developments in AI interest, and sensing and sensor technologies that are advancing so quickly. So those are the, I think, capabilities and technologies that I am seeing compared to 10 years ago. But in terms of the concepts and the possible techniques and strategies, especially in a location like Jeddah, managing high humidity, especially during the nighttime. Daytime somewhat still challenging, but nighttime is further challenge. So using dehumidification systems with chilled water and cold water if available. If not, can be generated through, again, engineered systems. Maybe using solar energy is one of them. Also, other systems. Back then, desiccant systems was an approach discussed. So some of those discussions actually led to several graduate student research applications and some developments. So I was very pleased to see that. And again, super interested and excited about this opportunity to collaborate. I think there are possibilities when considering, our locations in arid desert regions. There are similarities in Saudi Arabia as well, especially in inland locations. But again, we need to consider the technology and suitability, adaptability to local needs, the consumer needs, the economics, the finances, the climate, I think, and combining all of our expertise here, I think this will be an exciting kind of route to explore those possibilities.
We just have one quick question from Gabby, and then we'll move on to the next, talk by Mark. So Gabby, can you ask your question?
Yes. Can you hear me? Yes.
Yes.
Yeah. Thank you for the talk. That was quite amazing, especially for me, I'm working in the open fields and in greenhouses, and I'm traveling around the world. How do you see the world in 2050? Are we gonna grow greenhouses instead of plants in all the nice environments, which we losing a bit of green? I know it from Thailand and Vietnam. It's already completely full of greenhouses. So I know for Saudi it wouldn't matter so much, but environments where it's still nice and green, are we going to lose a bit of our nature and growing just greenhouses in the future?
That's a great question, Gabriela. Thank you so much for asking that. At the early on in my, starting in my presentation, I kind of wanted to give a message saying that, I think towards the future, we will see integrated systems. It's not going to be one over another. There's no, again, single bullet solution. It will be integrated, adaptive solutions to local needs and considering the resource availabilities. We will continue growing in field-based agriculture systems. We will need to consider greenhouses and vertical farms. In the past year and a half, as we've been going through a pandemic, it brought different challenges. Now, with the field growers not being able to ship their produce to long distances, not having access to labor accessibility, availability, consumers' interest into local production, safe, nutritious food, fresh food, interest, and decentralized food production and presentation to the consumers. These are now all interest also for traditional growers and farmers. They're not going to give up on their traditional way of growing things, but they will integrate in greenhouses and vertical farms. So I think we will see combination of technologies, systems grown. Again, each location region is unique. I have a colleague who knows the plants biology, plant needs very well. A vice man recently told me that it's so hot here, water is not available. And I see that the only is capitalized to grow crops sustainably, sustainable agriculture is through perishable foods, through controlled environment agriculture. So again, locations makes a huge difference. It will play a role, but we will see these systems working together, complementary fashion, in a circular fashion, to address our needs.
Thank you very much. Yeah, thank you very much, Murat.
Thank you very much for having me again.
Okay, let us thank Professor Kasira for this fantastic talk. And so next, then we have the second talk by Professor Mark Tester. So Mark, are you ready? Can you share your screen?
Yes.
Okay, then I have to introduce quickly my neighbor first. Okay. Yeah, so Professor Mark Tester is the principal investigator for SALT lab. So these are the salties and the CDA. So Mark, is famous really for, his work in understanding the molecular underpinnings of salinity tolerance in different plant species, and apply and translate these actually knowledge into improving the performance of crops like wheat and rice. So a few years ago, he changed it. So he said, well, let us just do the opposite way by domesticating plants. So he's now focusing on three major crops like his quinoa and the Salicornia and the tomato. So he published really quite impressive work by showing and investigating also different aspects of quinoa, and of course publishing the genome sequence of quinoa in Nature in 2017. And he is now focusing on translating this knowledge into really products, which is quite impressive. He co-founded also Red Sea Farms with Ryan Leffers. So this is really showing the power of knowledge when it's translated into products. Last year, Mark was the head of the food sector in Neom. These are the impressive flagship city of Saudi Arabia, high-tech city of the future. And in 2018, 2019, Mark is a highly cited researcher. So he gets probably around or more than 5,000 times of citations per year. That's also quite impressive. And he has won the Khalifa International Award for Distinguished Innovative Studies in Agriculture and Modern Technologies. And he's honorary professor at the University of Adelaide in Australia. So Mark, you have the stage. Please share your screen. And I ask everyone, please turn off your microphones and give your full attention to Professor Mark Tester.
Thank you very much, Magdi, neighbor. It's lovely. Look, it's great to have this chance. And I think this initiative led by Rod is it's really fantastic to form a collaboration with University of Arizona. And there's so many commonalities, as Murat just so nicely described in the environment. I think we could do a lot with each other and learn a lot from each other. So I'll just very briefly run through me and what we are doing in the SALT lab by way of introduction. And I'm a plant physiologist, if you want to describe me as anything, but more generally, I'm a plant guy. I taught in Cambridge for over 10 years, and I taught a lot of different courses, and the best way to learn is to teach. And I have become quite a generalist. More recently, of course, collaborating with Ryan Leffers, an agricultural engineer, and really broadening out into the systems approach. And my experience at NEOM really sensitized me, made me very receptive to the importance of engineering and a systems level approach for trying to develop sustainable agricultural systems. So there I've got sort of a summary of some of the things that I've done in my path as just a human, as a guy, starting with cell biology as a PhD student, through to really adopting forward genetics with some vigor during my time in Australia after Cambridge. I built the Plant Accelerator as an automated phenotyping system. It's very, very early, one of these types of facilities. And that was really to help me try to bring phenotyping into a level of automation, which might help us physiologists catch up with the genomics people, so we'll be able to do real genetics with a lot of different traits semi-automatically. That type of facility, I think, really is the right thing to do, and in the field as well, of course, in particular using drone-based imaging technologies. And yeah, it's a pleasure to be, I think we're in a very exciting time in plant sciences, because I think these phenotyping technologies, genotyping technologies really are really ready to come together, and we can have an opportunity to do things like genetics in quinoa, where just a few years ago, there was no automated phenotyping and no genomics and no genetics. Or very, very little genetics. So the aim of our work is to try to develop saltwater-based agricultural systems to improve the sustainability of food production systems. And the vehicle for truly testing that under the torchlight of economic, not just environmental, but economic sustainability is the company Red Sea Farms. So the context of some of our work is that a lot of the world's food is produced under irrigation. In fact, 2/3 of all the water that humans use is used for agriculture, and every aquifer that is being used, every major aquifer that is being used for irrigation is being depleted. So we are unsustainably mining our water resources for a large fraction of our food production. It's most spectacular in the Punjab, but also very severe reductions in many areas in the Middle East as well. So we have to change. And one way we can change is to reduce the pressure on our freshwater resources by substituting freshwater with water resources that we currently do not use, ones which are so massively abundant on the planet, brackish water and seawater. So we want to unlock seawater and other water we currently cannot use to try to help feed the world. If we're going to unlock something, then we need a key. The keys I've just been describing, the genomics and the phenotyping, high throughput phenotyping. This provides us with serious opportunities, very big opportunities for very serious innovation, and particular opportunities to unlock wild germplasm. So I want to unlock wild germplasm, both to make existing crops more salt tolerant, and as Magdi described in his introduction, to get salt tolerant plants and see if we can domesticate them, turn them into crops. An example of some of our older work where we were trying to make existing crops more salt tolerant was with durum wheat, where Rana Munns discovered a line in a screening program that had much of durum wheat that had much lower sodium accumulation in the shoot. It turned out this was derived from an old cross between an old diploid relative done by a PhD student back in the 60s, and they'd accidentally brought in a gene from the diploid wild relative, which had been lost during the domestication process of durum wheat, which was helping exclude sodium from the shoot. And this led to very significant increases in salinity tolerance, although it should be pointed out only in the higher saline areas of the field where the yields had been reduced by 2/3. So it was a nice bit of science, proud of the science, proud of all the work that we did, but its true impact is really quite modest, because it was really having an impact in only a small fraction of the fields. We're still doing some of this work, though, trait-based improvements of existing crops, building on Nadia Tamimi's PhD. Dongryong is now characterizing genes in a locus where she was, that Nadia characterized, where the plants were better able to maintain transpiration use efficiency in saline conditions relative to non-saline conditions. And Dongryong is zooming in on the causative gene within that locus. There are 25 candidate genes, including 5 tandemly repeated protein kinases. We're also working with tomatoes, as Magdi mentioned, in particular, trying to develop rootstocks onto which commercial scions can be grafted that would improve salinity tolerance. So these are some of the areas where we're trying to improve the tolerance of existing crops. But yes, a few years ago, I was starting to realize that if we're really going to make big step changes, we need to be getting salt-tolerant plants and domesticating them. And sort of as a practice run, we're working with quinoa. It's already partially domesticated, but certainly not fully domesticated. It has a pretty good salinity tolerance. It's not working it doesn't grow in seawater, but it can grow in half seawater and produce a reasonable crop. So it's not bad. It's definitely a crop for marginal lands. It's grown, of course, in the Altiplano in areas such as Bolivia, photographed here, and where the bloke will say, we have nothing else but quinoa. It really is a tough plant. And it also has huge potential. It has very high crop yield potential and also very high nutritional value. I do think of it as a potentially useful crop for climate change in the future. So to try to develop quinoa, one of the fundamentals is to sequence the genome, which we did, and I'll show you a slide in a moment, a bit more on that. And we've resequenced a large number of lines as well to form a platform for genetic association genetic studies to try to improve things like early vigor, branching, herbicide resistance, day length insensitivity. Heat tolerance is a particular project of interest, and especially in this area, the plant is sensitive to heat. It's one of its weaknesses. And Elodie has put in a grant, and she's already done quite a lot of experiments with the wild relative of quinoa, Quinopodium hircinum, which seems to have very high levels of heat tolerance. Elodie has, in collaboration with David Jarvis in particular, really greatly improved the quinoa genome that was published 3 years ago in that work, senior authored by David Jarvis. And she's really done a fantastic job. I'm not going to go into details. I'm just introducing to the fact that we have done this. And Elodie and the guys in Brigham Young University have also been sequencing some of the diploid progenitors, close relatives of quinoa. And there really is very nice synthetic alignment to the respective subgenomes of quinoa. Elodie's done a great job with this. Resequencing then starts to enable you to look at population structure. This was a figure put together by Dylan Sarange, where it looks like the quinoa population, at least the 1,000 lines that were sequenced, fall into 7 main groups. You'll see the number 8 there, but that's because one of the groups are the wild relatives, the outgroup, with lowland and highland quinoa falling apart really quite distinctly. And this is, of course, providing us the background for doing quantitative genetics, which we're trying to do in the field using traditional and new phenotyping technologies, and doing this in a lot of different sites with collaborators around the world. In KAUST, I just really want to acknowledge the existing quinoa team, fantastic group of people working really hard on this, what I find very exciting work. Got collaborators scattered around the world. Not all are growing 1,000 lines by any means. Some are only growing a few, some are growing a good population. We're trying to develop a core set, which really is representative, or a set of core sets, which are representative of covering the genetic diversity that you saw a couple of slides earlier. We need to phenotype them using what I would call traditional methods, and Gabi and Clara have been leading the development of a global standard in collaboration with CIRAD and FAO to have a field-based, cheap, accessible, readily accessible for developing countries where a lot of this technology is so important, global standard phenotyping system. That's great. And of course, that can be complemented by high-tech phenotyping, such as being done here in collaboration with Matthew McCabe and Jocelyn Lopez and Casper Johansson. They're in Building 4 and doing fabulous work, flying their drones and using advanced imaging, computer vision, AI-based methods for analyzing the plants in the field. In a similar vein, I'd just like to point out or highlight Mariam Alia's PhD, where she, and with really good input from Magda Julkovska as well, were phenotyping a large Arabidopsis population and looking at a lot of different photosynthetic traits, and actually getting some very, very interesting results, which were just published. They came out properly in Plant Journal just a week or two ago. So have a look out for that paper. But Mariam, using these automated methods of plant phenotyping really was gathering huge numbers of traits and huge numbers of lines. And one of the big issues really is simply, in inverted commas, the data management and processing. And the students in the lab got together with Magda and developed this app, which I really commend to people, both in KAUST, they probably know about it, but also in Arizona, called MV app, for multivariate analysis app. And it really has a lovely set, very clear, very user-friendly for plant scientists to be able to handle and analyze these massive data sets that we now can so much more easily generate. This was published in Plant Physiology. Now it's almost a couple of years ago, a year and a half ago. So we can phenotype, genotype, put this together to do genome-wide association studies to try to identify loci that are contributing to the phenotypic traits being studied. So for example, here, Clara, I don't know, was this, yes, Clara did this analysis, sorry, Clara, where she was looking at plant height and looking at some loci are sensitive. So when you grow plants in long days or short days, so it has sensitive to daylight, a lot of quinoa. You will find some loci are stable and others are not. And of course, this now needs to be drilled into to characterize the candidate genes in these loci. And Dylan Sarange, we had a really good collaboration with the University of Kiel, and Dylan Sarange, the PhD student, Christian Jung was the professor there. Fantastic work using the populations that and sequencing that we'd provided. And he's identified nice loci with very strong candidate genes for things like flower color and saponin content. And he's sort of almost using that as validation for looking at other traits for which there's pretty much nothing known in quinoa. And here's one candidate, locus, well, it's not a candidate locus, but locus containing a nice strong candidate gene, which has got pleiotropic effects across several different plants and plausibly connected plant traits. So this is the type of work that we're trying to do in quinoa by way of introduction. So that's good, but you know, you've got quinoa and half seawater. If you're in an environment like Saudi Arabia, half seawater, that means you still need half fresh water. Where are you going to get that from? You're either going to mine it unsustainably from the groundwater, or you're going to use desalination. Neither option is sustainable. One's not environmentally sustainable, the other is not economically sustainable. And really, if we could develop crops that can grow in full seawater for an extreme environment like Saudi Arabia, I really think that's in many ways the only way to go for long-term environmental and economic sustainability in extreme environments like we find here. So Salicornia is one of these remarkable plants that grows very well, even in full seawater. And it's got lots of phenotypic variation, gross morphological, as well as yield and harvest index. Octavio Salazar has done a draft genome for 3 species of Salicornia, including Bigelovii, which is tetraploid. So that's a tricky one. It's an autotetraploid. But also Europea, he's done a good sequence for, as well as the Indian Salicornia, Salicornia brachiata. And Octavio did a, so I'm just keeping an eye on the time. I thought I'd better go very fast. So here we go. So Octavio did a proteomics study, because we're not just trying to do very applied work, we're also doing work which is of fundamental scientific interest. And the really noticeable thing about Salicornia, besides its lovely oil-rich seeds and phenotypic diversity, is that it can accumulate 1.5 molar sodium chloride in the shoots. That's incredible. I mean, rice will curl up its toes and start going crinkly when it's got 1/100th of that concentration. Barley, which is a pretty salt-tolerant cereal, that'll start going crinkly and turning up its toes with 1/10 of that concentration. So Salicornia is way off scale in terms of its ability to tolerate, grow, produce seed, produce yield with this massive concentration of sodium in its accumulated in its above ground parts. And so, you know, that has to include, must be central to that, must be extraordinary abilities to sequester sodium and chloride in its subcellular organelles. So let's have a look at the Organella Transport. And very interestingly, the Salicornia tonoplast and a sucrose gradient comes out at a much higher density than the plasma membrane. That's the reverse of normal, if you like, plants. So already there lies a hint. We guess that we propose that this high density of the tonoplast is because it has a higher concentration of proteins, perhaps involved in the subcellular sequestration of the sodium and the chloride. So Octavio did proteomics on this. The subcellular sequestration, there's a little bit of evidence suggesting that it is inducible by salinity. So Octavio did the proteomics, and he did it in salt, low salt and high salt treated plants. And it's really beautiful work. And here, I love this graph that Octavio generated, where you've got a Pca analysis with the identification of the proteins which are with a high probability of being the plasma membrane, high probability in this case in the mitochondria, the blue, and then look at these ones out here. There's the vacuolar ones in low and high salinity. So he was able to identify about 151 proteins with high confidence from the tonoplast, looked at those that were inducible, and then cloned 46 of these and started to characterize them in yeast as a heterologous expression system. So we're looking here at a yeast which has got some of its sodium transporters already knocked out, and then so they're very salt sensitive yeast, and seeing if he was able to alter their salinity tolerance, and did find things. There was one protein here and a transporter, NHX3, and these are now needing to be characterized further. This is work which we're still ongoing in the lab. So we're trying to work here in making some crops for open fields, but I think unless we can develop plants that can grow in seawater and also have very high temperature tolerance, really, and I still think that's a good thing to do, by the way, I should emphasize. But I think Murat said it very well earlier. In fact, he said lots of very good things earlier. It's nice coming after you, Murat. The best option, I think, for sustainable agricultural activity in these types of extreme environments where there's very little freshwater availability is to use controlled environment agriculture. I wouldn't say it's the only option, but it's certainly a very good option, a very strong option. Furthermore, it's only economically viable to grow a small subset of the foods that we need in covered environmental agriculture. So don't worry, Gabi, there still will be green fields with wheat blowing in the winds, just not in Saudi Arabia, okay? But we actually need, by many analyses, not more starchy foods and not more starchy foods that we're feeding to animals to be killed for red meat production, but we actually need, because the global average, we eat way more starchy foods and red meat than we need. We're already producing enough on the planet for people to be healthily fed even by 2050, according to this analysis from the Eat Lancet Commission. It's a very credible analysis. But what they do show very clearly is what we need is a lot more production of fresh fruits and vegetables. And this really is the primary need for food production in the world over the next decades, many decades to come. So I think it's quite good to be focusing on covered environmental agriculture because of its sustainability, its lower environmental footprint, at least for fruits and vegetables, and also because we need more fruits and vegetables unequivocally to be produced over the next decades. And so I was very lucky to be able to form a partnership with, yes, Murat, an agricultural engineer, Ryan Lafers, and we're able to put together the plant science and the agricultural engineering to try to develop agricultural systems that are based on salt water, preferably ultimately seawater, and which are both not just producing food with a lower environmental footprint, but which are also economically viable. And we thought the best way to truly test the economic viability of these new systems is to form a company and see if it survives. So the first step in the company is to, this is how Ryan and I started the company, was to grow salt-tolerant tomatoes using diluted seawater, all right, in a greenhouse cooled with seawater, big environmental impact, and where the roof, you have to clean the roof a couple of times a week in this environment to maintain production at high levels because of the dust. So you have to clean the roof a couple of times a week. That uses a lot of water. So if we could develop a system in collaboration with Oasis, the solar panel waterless cleaning company, clean the roof with no water, we would end up developing a system where about 80 to 95%, depending on the temperature, humidity, and so on, 80 to 95% of the fresh water is replaced with seawater. That's a pretty good system. So what we're bringing together is the plant science and the engineering and technology with, and again, Murat identified this perfectly in the question and answer session, with IoT and AI and controlling it with our own in-house developed smart system to try to have greenhouses growing crops, horticultural crops, high-value horticultural crops, with as little fresh water as is economically viable. This is just a table giving summary of the of some of the numbers, and the key thing is that what we're doing, we think, is breaking this food, water, energy nexus that most production systems in the world are trapped in. You can reduce the amount of water by having, for example, a mechanically cooled greenhouse, massive saving in water, but that comes with a massive cost in energy. And then a normal greenhouse, of course, uses quite low amounts of energy, but with a massive cost in water. And we're proposing a system by substitution of fresh water, seawater, especially for the cooling, where we're saving with both fresh water and energy consumption. Okay, Red Sea Farms has since developed a lot and also merged with another company called Iris, which has, yes, Murat, you mentioned them as well, transparent to the photosynthetically active radiation photovoltaic cells. So photovoltaics are absorbing in the infrared. And the first tens of square meters of these are being put onto our research and development greenhouse here on the KAUST campus, as in the Red Sea Farms one. We're now developing a whole stack of different technologies to try to reduce the environmental footprint of food production, in particular in covered environments. And this is just a final slide where, in the spirit of trying to explore, I was just throwing a few things out here, looking at where we might have CDA, University of Arizona collaborative opportunities just in the little area that my lab works in, and Red Sea Farms, I should say, also works in. So in open fields, we've got grafted tomatoes that I introduced the concept to you earlier. Perhaps we can do field trials in the Arizona environment to complement the field trials that we've already got underway in northern Egypt at the moment. There's also, we've got this huge international consortium for quinoa, which is now likely to be funded through the EU and the FAO, and wondering if there's anybody in UA with an interest in in quinoa. In the greenhouses, of course, with Murat, it would be wonderful to be able to collaborate very actively on that. And in fact, we're already booking a discussion later this week on that, Murat and me. And then, of course, in the lab, we've got a lot of genes of interest from the forward genetic work. We just don't have enough time to characterize all of these, although we would love to. And I'd be very happy to discuss potential collaborative opportunities there as well. And I'd just like to end after 26 minutes with just thanking a huge number of people. It's a crazy slide, really. It's a wonderful collaborative world we live in plant science. I think it's fantastic. Of course, I would love to thank the people in my lab. I've been acknowledging the work from individuals, especially postdocs and students, as we've gone through. But I'd also like to particularly acknowledge the three technicians in the lab and in the greenhouse: Noha, Dinara Hongwei, with Gabi, of course, but she's been named on a couple of slides. But these three women have really, their inputs is fantastic. But I'd just like to thank my lab very, very warmly. Of course, now this is how we meet. Isn't that sad? But I'd like to take an opportunity here to have a photograph with this lovely smile with Vanessa. I'd just like to thank her in particular as the research scientist, in effect, my deputy and the person who really does a lot of the work running the lab now. I'd like to thank you all for listening as well. Thank you.
Thank you, Mark. As always, it's quite impressive. Yeah.
Thanks.
Okay, any questions, guys? We still have time for questions. Questions?
Hello, can I ask a question? For the cooling of the greenhouses with the seawater, that's a really interesting technology, but how do you keep the salt crystals from forming within the system? Don't you have to clean it as well at some point with fresh water?
Absolutely, Katja. Yes, absolutely. So again, with these broad principles of IoT, we have sensors in the water sensing the temperature, concentration, and some of the ion composition. And we have what happens is actually it's quite a nice system. The guys in Red Sea Farms developed this to bleed in water as the conductivity goes up to maintain the concentration of salts below a threshold. And then, of course, every so often, you can't bleed in more water because it's there's too much salt in the system, and then you flush it. So Ashley Persglove in Red Sea Farms developed this, and it's quite nice to watch it work. So yeah, you're absolutely right. There's another thing I would mention is that there's also a possibility of pre-preparing the saline water that you're using to be able to go to higher concentrations by removing the divalent cations using nanofiltration. So this is a system which might sound a little exotic, and it's certainly not widespread, but it is used in some engineering applications. And so we're thinking that this could also really help with exactly that issue you're raising, Katja. So at the moment, we're doing it through monitoring and management. And yeah, when we get our R&D facility operating for many, many months or years, then we'll be able to tell in practice how good that is at reducing precipitation. But in parallel with that, we're also going to develop a system with nanofiltration. We're in discussions with pretty advanced discussions with a collaborator for that.
Can I ask another question, maybe follow up? This is more about the genome part that you mentioned. You found new plants that are growing completely in the salt water. And can you identify new pathways, how they, or are there other ion ponds in those plants to extract the salt components? And can you transfer this to crop plants maybe in the future?
Yeah, I love that last bit of your question, because that was certainly part of my original driver. But I don't know, using genetic modification to deliver research outputs, it's so depressing. I mean, Brander Wolf is fabulous because he's such a great champion and still fighting on, and it may, we may well win. I don't know if Brander is here and would like to add a comment on top of mine, but at the moment, yes, I like I like what you've said. And yes, we've found, like I showed, NHX3 seems to be one of Octavio's candidates. There's quite a lot of candidates actually, and we're quite, we're trying to be as agnostic as possible. Let the plants tell us what the candidates are. I'll tell you one other gene, which seems to be, because we're about to submit the paper, which seems to be in the tonoplast, in the vacuolar membrane of the Salicornia, and it's SOS1, which is a sodium proton antiporter, which has been previously identified to be at the plasma membrane, and it's quite possible that in Salicornia it has been relocalized, it's been diverted to the vacuolar membrane, and it's now sort of been neo-functionalized, if you like. But testing that hypothesis is very difficult. We can't make transgenic Salicornia. We need Magdi's magic and probably more to see if we're able to maybe gene edit it in some way to test this hypothesis. But yeah, I think there's quite a few candidate genes, Katia. And yes, I would love to be able to deliver this through a GM pathway into crop plants. Wouldn't it be wonderful? You grow rice with this type of subcellular sodium sequestration power. That could be massively impactful. But Branda, can we deliver it?
Branda has a question, actually.
I have a question. Well, first a comment. I hope I'll convey in a few weeks when it's my turn to talk that I think I very much hope we can deliver on GM, and the motivation is still there. Yeah. So perhaps because I've still got 20 odd years before I retire, I'm still.
Yes. Thanks for reminding me, Branda.
Well, maybe you'll keep on going, like Jonathan. Anyway, Mark, I have a question. With the evaporative cooling system, I guess the relative humidity is very important. So here on the west coast, it's currently 54% relative humidity, whereas in Dammam, on the east coast of Saudi Arabia, I think it's 18%. So there's quite a big difference. Would that make it significantly cheaper and easier to grow your crops in Dammam on the east coast?
Yeah, actually, you only have to go not that many tens of kilometers inland, and the humidity crashes, goes down very, very quickly. So Rod was in Taif a couple of days ago, but it was a lot less humid there than it is here.
But you wouldn't pump seawater all the way to
No. But there is a lot of salty. No. Seawater is sort of the ultimate, but for every drop of fresh water underground, there's a drop of salty water. And you can use brackish groundwater as well, as long as you have systems for the disposal of the brine. But farms are all, if they're using water, like we see farms, it makes you weep, Brandon, where they've got their great big, they're pumping out the brackish water, they're desalinizing it, and it goes straight from the desal unit onto the evaporative pads. Yeah. So don't give me any, we've got to get rid of the brine problem. Therefore, the system's so spectacularly inefficient and dumb already. So this is where we're really, I think we can make a big impact actually with greenhouses inland. But I'm sorry, I'm ducking the question. For humidity, yes, we are now building a greenhouse about an hour north of KAUST. Well, we've got the pilot at KAUST, 2,000 square meters, but we're building 10,000 square meters with the aim of that expanding over the next 3 years to 100,000 square meters, about 1 hour north of KAUST campus. And it's within 300 meters of the sea. And Murat again put his finger on the technology using liquid desiccants and using solar powered chilled liquid desiccants. From Ryan's PhD work, here at KAUST, that Murat also alluded to, it looks really very promising that, and from actually our trials in the last 6 months within Red Sea Farms, it's looking really promising that we will be able to cool greenhouses even with extremely, like 100% humidity, because you wait for new people here. This is nothing what's happening in KAUST this month. In about 2 months' time, you'll have the condensation on the outside of your windows at night, because it's actually saturation humidity at night for 1 or 2 months. It's impossible to evaporatively cool a greenhouse. But with liquid desiccants and with them chilled using solar, we are thinking we can have a system that will work technically, and then it's the economic viability of that system that we're now testing. Will be.
Thank you, Mark. And I think this is really quite impressive. I see Rad is with us right now. So Rad is the champion of this joint seminar series. So Rad, you want to add something? To have some closing remarks on these great seminars?
Yeah, I apologize for. I started this thing and I didn't show up, so it's anyway. Please forgive me. Was out in the field today. Pardon me?
Yes. You've been doing science,
exactly. So, no, this is great. I mean, it's we had another really good turnout, and
yeah,
it's I think there's so many opportunities that we can have between Arizona and KAUST. So it's I really want to thank my good friend Murat for coming, and all my Arizona colleagues in the audience, and good to see you all, and really great talks. I caught about half of your talk, Murat, and I got all of yours, Mark, and so I think we just got to keep the communication going, and we now have Rebecca joining us hopefully in a few months, so that we have a direct link now on the CEA side, and yeah, it's exciting. So so I did have one question for Mark. What other vegetables are you experimenting with respect to graphing for salt tolerance? Is it only tomato?
Yeah, on the grafting side and the development of salt tolerant rootstocks, it's only tomato. That's hard enough. But it's a very, very nice question, because there's almost all melons grown in the world are grafted. It's an incredibly high proportion of melons grafted, and melons are very salt sensitive plants. And I've been starting to eye off, I have to admit, I'm starting to eye off a couple of these very tough melons that are sitting out there in the wild. But I'm completely, that's another whole research program, and I just don't have the resources at the moment to go beyond.
Okay.
Yeah.
And I had a question for Murat too. Murat, I mean, I don't know if you explained the CEA program at Arizona, but how many students do you have? And because we're talking at KAUST, we're really talking about really promoting control CEA across the whole nation for fresh vegetables, and we need to figure out a way to train these people, and I think there could be some definite exchanges between our programs in that realm.
Yeah, absolutely. So in my lab, I have about 6 students right now, and in the program here at the CEAC, graduate students, I think we're close to 15, and that number is going to increase. Our undergraduate students with CEA emphasis, I think, through the Sustainable Plant Systems program and the BE Biosystems Engineering. I think that numbers is somewhere around, I think 30, 40. But a lot of attraction, interest towards controlled environment agriculture, urban agriculture, indoor agriculture, and indoor farming. I believe, as I mentioned in my talk, our youngsters, our young minds are really interested in ag-tech and technology, data, so I think controlled environment agriculture gives that excitement and provides, opportunities for them. So, and we need our students in this program because they are in high demand by the industry. Yeah. Trying to keep up with the demand coming from the industry. Yeah. That's exciting. But I think they will need, we need to educate them in a way that they need to understand both the biology, the plant requirements, as well as the engineering. And those students, graduating with those skill sets are better positioned, strongly positioned in the marketplace.
Yeah. I was going to say that this ain't grandpa's farm anymore. You know, it's with all what, you know, you can't just be growing plants. It's all the technology and stuff. It makes it pretty exciting. So,
Right.
yeah, well, let's keep the dialogue coming. You know, we have, I think, 5 more pairs of talks, and again, a great turnout. So thanks a lot. Please, everybody come back. We're going to have an exciting set of talks next week. And I guess with that, I want to thank Magdi for being.
We
and
thank the
I'll be back next
speakers.
week.
The speakers.
Yeah, thanks to Maddie as well. Thanks, Rod. Thanks for leading this effort, and thanks, Maggie, as well as your team there, Robin, for you know, helping us with this. So, and everyone joining today and attending the webinar. Really excited. Looking forward to developing the collaboration. So.
Okay. Well, next time we'll have you come out. Okay, and then we have to come visit you guys too.
Yep, that cafe next to Beacon is wonderful.
Yeah,
I really enjoyed that setting there. It's beautiful.
Yeah, just don't come out this time of the year. So, okay.
Right.
All right. Thank you, Mark, as well. Thanks.
Okay,
Thanks,
thanks,
Maggie.
Rod.
Thanks, everybody.
Rod, could you stay on for a minute?
Yes, I can.
Thanks.